I'm Anjani. I'm lead content director for drug discovery chemistry. And I'm here today with Gottfried Schroeder from Merck, who is our featured speaker on the small molecules for immunology and oncology and COVID track, which is taking place at our event, Drug Discovery Chemistry Virtual. So again, it's virtual this year. It's taking place May 18th to the 20th, the whole event. The small molecules for immunology and oncology track is in the second half of the event, May 19th to the 20th. And Gottfried is going to give us a preview of his talk to hopefully entice you all to join. You can, of course, see all the other great speakers and talks we have lined up by going to our agendas on the web. So Gottfried is going to be speaking about STING, which is a molecule inside the cell that plays roles in innate immunity and is quite hot in industry. Like we know Nimbus is working on it, GSK, they spoke about it last year. We also have Silicon Therapeutics speaking about STING, and STING is a unique molecule because it has a role in both cancer and autoimmune and inflammation. If you modulate it one way, it's to affect or ameliorate inflammation. If it's in another way, it's for cancer. So Gottfried, I know you've spoken about STING before at our 2019 event when we were in person in San Diego. So what is different about your talk this year?
Right. Well, first of all, I want to thank you, Anjani, for this opportunity. And I would say the similarities end with the target between the two talks. My earlier talk actually focused on first-generation STING agonists, namely cyclic dinucleotide analogs of the known STING ligand, cGAMP. In this presentation, I'll describe a non-nucleotide STING agonist with a radically different mechanism of action, highlighted in our recent publication in Science late last year.
Great. So what is so interesting about STING, or why the buzz, as I alluded to before?
Right, well, a lot of it circles around the field of immuno-oncology. Recent clinical success with new cancer therapies that block immune checkpoint pathways, such as antibodies to PD-1, have led to simply intense efforts focused on other immune pathways that may be modulated to enhance the therapeutic benefit of checkpoint inhibitors. The therapeutic potential of STING agonism in particular has been demonstrated previously in preclinical mouse tumor models, both alone and in combination with PD-1 blockade.
And why is STING hard to modulate?
Well, I'd venture to say that modulation of STING itself isn't really the hard part. Indeed, analogs of cGAMP are potent ligands. However, when administered systemically, such agonists induce inflammatory cytokines both in the tumor and in non-tumor tissues to the same level, owing simply to the ubiquitous expression of STING throughout the body. This, unfortunately, effectively relegates their application to a narrow set of tumors, ones that can be dosed by direct intratumor injection. So, to instead address a broad spectrum of tumors, for instance, those found in internal organs, the true challenge is to find a tumor targeting agonist suitable for systemic administration.
So what about your work makes it easier for systemic administration?
Right. So in fact, we have identified a previously unknown compound, designated MSA2, that exhibits such behavior through its distinctive mechanism of action. Moreover, MSA2 is amenable to oral administration, a desirable delivery route because of convenience and low cost. Indeed, orally dosed MSA2 is well tolerated in mice and exhibits STING-dependent antitumor activity, both as a monotherapy and combined with antibodies against PD-1.
And what aspect of STING, modulating STING, will your work focus on?
Right. My particular talk is going to focus on the properties of MSA2 and its unique mechanism of action. Extensive biochemical and biophysical experimental analysis indicates that pre-dimerization of MSA2 in solution is actually required for binding to STING. In addition, cellular potency increased upon extracellular acidification to mimic the tumor microenvironment. These properties appear to underpin the favorable activity and tolerability profiles observed for this particular compound.
Great. Well, thank you. We look forward to hearing more. So I hope you all can join us again, May 19th to the 20th. And then next year, 2022, we'll hopefully be back in person in the spring in San Diego.
Sounds
So
great.
thank you. Hope to see you all on screen, and good luck with your work. Bye.
Thanks again. Bye.